Welcome to a new SciPod. This podcast explores the essential roles that advanced science plays in our lives. In every episode, we break down complex scientific discoveries and share professional insight on the real world and on the real-world relevance of research. Join us as we go through discoveries that fuel curiosity and inspire for further inspiration. Welcome aboard a new SciPod. My name is Balkiya Ospan, and I am a second-year biological student.
And my name is Amanbek Bekturgan. I'm first year life science master student, and today's episode features Dr. Sarbassov, a leading researcher who has discovered the critical cell pathways and is developing groundbreaking cancer treatments. His works has significantly advanced the understanding of the mTOR pathway, which stands for mammalian target of rapamycin, and we will cover that a little bit later in the podcast. This pathway controls cell growth and survival. Professor Sarbassov has spent 27 years conducting research in the USA, including at MD Anderson Cancer Center, one of the world's most respected centers devoted exclusively to cancer patient treatment, research, education, and prevention. In 2019, he returned to Kazakhstan, and now he serves as director of the National Laboratory of Astana at Nazarbayev University here. And he also holds the position of the vice president of the National Science Academy. Currently, he and his team continue to develop effective cancer drugs that are undergoing clinical trials on people.
Yes. Can you imagine that every second, and even now as you're listening to us, your cells are going through mechanisms which determine their fate? Those mechanisms are called signaling pathways, which will be the topic of our discussion with Dr. Sarbassov. So enjoy and learn something new with us. So, Professor, nice to meet you. We are honored to have you in our podcast. So, how are you feeling today?
Barmısız ba, hayırlı keş. Thank you very much for inviting. It's my first podcast in my life, so I really having great time. It's perfect setting for podcast.
Thank you very much. Back in the 80s, science was not a common choice for career paths. Can you tell us what made you decide to study science?
That's a question that I can talk hours about. But technically, I grew up as ordinary person. I was not super. I didn't have any super strong capability during school. It just happened maybe mostly because of family input, because my mother was teaching biology and geography at school, and my father was a scientist and was teaching at agriculture university. So, and he always was talking about science, how important science for society. So, and during childhood I didn't pay much attention, but because I thought scientists are some super people, super smart, and because my grades were not strong, and I was just mostly B, C, something like that. We call it Tikhtroshnik. Yeah. So, but it happens that in my life. So, when I joined actually university, and that what make big difference. So. Another factor, of course, is environment. So, but to get ready for environment, it took some time for me because when I entered Kazakh National University biology department, idea was I wanted to just work for some fish farm, just very specific. I was thinking just doing some very simple because I used to like to have aquarium at home, and I thought I'll just continue. That was my hobby, and I thought that's what I like. But later, of course, at university, I changed my mind completely, and because of environment. But the second thing was, of course, that you have to be ready. So, and I realized that when I started my school in 1984. At Kazakh National University biology department, I was still pretty maybe not mature yet. So, but then I went to army. So army really changed a lot because at that place, a lot of people dislike army. But I think army makes a difference because you went through military service and you become much more matured very quickly. And when I got back in 1987 from army, so then I decided, so I better do very well. If whatever I do, I try to do good. And at that time, I became a straight A student from this second year, and that's how it started. But of course, then most important thing, I joined. I was looking for something exciting because I thought I can do well now because I was more confidence went up, but I didn't know which area to focus on. And I changed 2 or 3 different departments. So I went first to botany, then I didn't because there was some idea that I could go to school to Moscow State University because they had some spots to send later for Moscow State University. But then I didn't like it at all because it was very descriptive science. And then later I went to physiology of plants. And but then when I met people at Department of Genetics and Molecular Biology, and that was very strong department when they actually did a lot of work in regulatory biology, and they were working on cell signaling. And I got luck, maybe it's a lot of luck as well. And cell signaling was so exciting because I was trying to understand how signaling pathway takes inside the cells. That was a very great attraction for me because I tried to understand how it works altogether, a secondary messenger system, like hormone regulation, and that was that's how I think that made a difference. And of course, again, so environment was critical, and that because science was already working at Department of Genetics and Molecular Biology, that led by our dean at that time, and of course, a professor chair of this department, and my teacher, Dr. Professor Rahmet Qazi Iskenderovich Bersimbayev. So, and I'm proud that now he's a distinguished professor at Eurasian National University, and we now continue work together, and we have we actually train students together. So that's my part how I became a scientist. Yeah.
Thank you
Wow.
very much. So you talked about that the biology department, like this department was very strong, and they were conducting research on cell signaling, etc. So could you describe how was science back then compared to now?
Okay. That's a good question. So science in 1980s, because I was saying I never wanted to become a scientist, but at school I decided I made big dramatic change in my life. And Professor Birsimbayev he actually strengthened our training by sending us for summer training to Novosibirsk State University. Technically, in Kazakhstan, of course, science was not well funded because it was all kind of leftover principle, whatever. So there was not much funding, and most of the science that was developed was mostly descriptive sciences, but then was not like high tech. There was not much high tech. And the only case I remember when it was our Academy of member is and president of Academy of Sciences is Professor Murat Aythojan, who actually was did his career, started his career in Moscow, and then he could come back to Almaty, Kazakhstan, and established the Institute of Molecular Biology. That was major breakthrough in 1980s, and a lot of young scientists joined and started working in that institution. I think it was one of the major breakthrough. But I actually started working at Kazakh National University under supervision of Dr. Bersimbayev. So, of course, that's what I'm saying, because when you compare what was science at that time, and you clearly, there is no doubt that, of course, now with today, actually science is getting much stronger. And we're still in the process of developing strong research programs, I think, but and Nazarbayev University is represents oasis when we, or I call it nucleation, nucleation of scientific development. I think we are developing at high rate, and I think, but still only now we generated critical mass that can develop into competitive science. So we're still in progress, and I think we just have to continue our focus on scientific development. And I think Ministry of Science and Higher Education and Minister Mirza Saisat Nurbek, he strongly supports our research programs, and I think things are not only at Nazarbayev University but through whole Kazakhstan. I think its time is we are in a transition to become strong, actually develop strong research programs, and I think that because it's critical for development of our country, so Kazakhstan. So I think things are moving forward, but I can that's only I think that becomes a priority, and I think within a short period of time things are changing, transforming into competitive science, and I think it's a critical point. That's why I think I'm very happy that I could contribute, or I'm in a still I hope I can do more in a long run.
Mhm. So, as you know, you had been conducting research in the USA for 27 years, working in one of the most world's respected cancer institutions. So, and also you established yourself as a leading researcher. So, what made you decide to come back to Kazakhstan?
Okay, so that's good. I better also give us a small introduction my scientific career in US. So technically, I got into US just because I started my scientific interest in Kazakh National University, because in my my senior year, we had this exchange program with because it was boundaries were suddenly opened for us because. And in 90, we started having this exchange program and went to US for 1 semester. So I went to the small private university in Arkansas, and from there I actually requested Professor Joe Jeffers from a small university. He actually had a friend at so it's in another luck maybe a friend faculty who was working at University of Arkansas for medical sciences, and he was actually one of the faculty who was recruiting graduate students for his program biochemistry and molecular biology. So when I met them and I understood which exams you have to take, and they actually accepted me later, and I started full-time as a student, graduate student in early 1993 already. So and when I graduated, and I was also lucky was joining Dr. Peterson's lab, who was working in understanding cell signaling pathways during muscle differentiation, and I was continue cell signaling studies, and it was pretty successful, and I felt like I did very good, and that's why. When I graduated there with 3 papers as a first author, and one of those proceeding on National Academy of Sciences, so all doors were open. I could go to Harvard, Cornell, or so. And then I chose Cornell for 1 year, Cornell Medical College, but I didn't like as postdoctoral training that I didn't like that research area, that immunology, cell signaling immunology. I was not exciting areas. No, at that time. For me, I mean, I didn't find it attractive, and then I switched actually to join the David Sabatini lab at Whitehead Institute. For Whitehead Institute, it's institute at Massachusetts Institute of Technology. So it's actually a top scientific center, and over there actually I worked from 1999 to 2006. So it was 7 years. That's where we're actually studying mTOR signaling. So and that's after successfully completing this our mTOR project in 2006, I got offered to join the MD Anderson Cancer Center. It's top 1 cancer center in US, but I think in the world. And over there, I work as a head of the lab for like 12 years, and then. Time was clicking, and I thought I crossed 50 years. When after 50 years, I realized it's because a lot of things were happening in Kazakhstan, and I was visiting Kazakhstan with lectureship, and then also I had my colleagues here, my friends here, so with whom I went to school in biology department. So technically, I made my mind that it's time to come back and do something important in your life, and actually pay back to your country. That's the main reason, yeah.
Yeah, that's actually a great thing, I think. So while you're working in science, I think when you have a lot of discoveries, you have also a lot of patents as well. Could you please describe the process and how difficult it is to obtain a patent, and how exactly does this happen usually?
Okay, very okay. So patenting is actually bread and butter for top universities, particularly at MIT when I was White Hat Institute. So they were patenting everything automatically, whatever, because they always discovered new things like new mTOR, like whatever new complexes, new technologies, everything. So there is patent office was automatically patenting straight. If it's a big finding, they were patenting every. So at that time, I was not really even well familiar with this patenting system because it was going by itself. I was not even involved. But later at MD Anderson, actually, they were always patenting something that were promising. So because they were maybe tight with budget, everything, so they only were patenting that that could kind of develop into the product or new methods or so. And when I met with the patent officers there, so at that place actually they were very stringent on selection of so you can of course submit your initial file for patent office, but they were selective and so on. So it's a whole process. I think that was his gap, I think, in Kazakhstan patenting system, because if we in the future long term we are planning to develop our competitive research programs, of course patenting will be important because it can bring revenues back. We're talking about not only local patents within the country, but we're talking international patents. So I think that's something we have to establish, this culture of patenting, how to do it right, because patent office is a very specific area when scientists become patent officers. So because they have to understand science and they have to understand marketing and also law as well. So it's all together. What was interesting was a patent I can discuss later. So they were very much helping me to develop our drug combination, because they were guiding like what type of patent can proceed and how to put it all together. I mean, it's a whole infrastructure that we have to develop in Kazakhstan.
So we cannot be like strict-minded and focus only on science, but also learn anything additional that also like has to do with marketing and etc. to develop and just to broaden our horizons of what we can do and think of. So it generally helps us to.
Your question is about patenting, or
yeah, about the patenting, like science and
patenting. I think we don't have to worry right now, because patenting will come when we become strong, and you have to patent when you discover something new and develop new methods. That's when you can patent. But I think it's time we better prioritize how to make a strong science first in Kazakhstan. So develop when we have our young scientists come, they're very motivated, they develop something something exciting, new technology, a new product, new drugs. I think it takes time, but we have to be ready. So my point is we have to understand we have to think about how to build this area, and we have to find the right people. So it's a creating environment to do research. First, I think focus to develop strong science, and when we have competitive science, then patents will start coming. We're not talking, because what I know, there was like 10 years ago or 5-6 years ago, the Ministry of Science required whenever you get your grant funding, they require to have at least 2 or 3 patents or 1 patent at least minimum as a result of your funding. So, and of course, everybody, you can comply with the requirements. You can patent local patents because there is no any screening, whatever you can write and say, because this is all definitely unique for Kazakhstan if you do something, because we are not competing with the whole world. And but that's why a lot of patents were coming, but they were not strong and competitive. So they're not like moving industry forward, but because they were required for successful completion of patent. But so it means if things come from the top, it doesn't. So they have to develop from the bottom. So it means you see my point. So And I think we're getting there. So now, what is good? So ministry learned a lot. So now this requirement is gone, and now we're just focusing on publishing good papers. And I think that's trends that is develop right, going right direction. It's matter of timing. So we don't have to be because quality is critical, and if we push for number, I think, of course, quantitative factor also eventually will lead to quality, but I think we have to understand that if we push too much for quantity, like publishing many papers, even Q1, it's not about just number of papers, because it's matter of what type of science we do. So we have to really focus on basic important questions. And my principle is, and what I learned at MIT, David Sabatin, when we discussed, he said, you have to consider any problem very seriously. Like if you like whatever you see some event, natural event, or any reaction. So if you don't understand how it works, it's very difficult. You have to consider it as a difficult problem. But as soon as you define this problem, identify the mechanism, things become simple and simple because you understand how it works. So technically, that's what I always tell. Everything is difficult, but unless you know how it works. So as soon as you know how it works, things become simple. That's what we are talking about. When you find something new, novel, new mechanism, of course, you can always find a way to apply it for everyday life.
Yes, I totally agree with you. I think we should not get scared of how complex things might look at the first glance. So basically, yes.
Yes. Also, as you know, you are General Director of National Laboratory of Astana. Could you describe the research potential of this institution? So you work here.
Thank you. So I think National Laboratory of Astana is, I call it, it's a leading research institution of the country. And it's truly because, first of all, strong scientists. And also we had tremendous support from universities. So we have top infrastructure. So, and that's why when I talk to my colleagues, I say there is you don't have any competitors, because you have a top infrastructure, and now you have to deliver. So, and I think what I can say, there are national awards. We have about 200 scientists. We have our own research administration that helps us quickly, dynamically set up all the documentation, because here we have a lot of paperwork with procurement, with reports, everything. So we have research office, we have administration taking care of this problem. So, and they're really helping us a lot. And when I when I became director in 1920s, I think 2020. So sorry. 2020, so October, I think. Yeah. So I realized there are also they accumulated a lot of problems. So I tried to change the system, that scientists will have much more time to do science instead of filling up reports or not too much paperwork, somehow to do ordering and so on. So and of course we also develop system how we can stimulate the young scientists, not just young, all scientists, researchers, because if researcher has sufficient funds, like not just one funding grants, but he helps to his colleague with another grant, so then they could have increased their salary, like, so it's up to 100%. So that's I mean, we develop a system that scientists have much more stimulus, or also that science is attractive for them. So, and I expect now that for all our scientists have everything. So now I say we have no excuse, and it's time to really show that we can deliver strong science. So that's a critical point right now. And besides, actually, we have very strong genomics. So our genomics lab is has most advanced platform. So it's Illumina NovaSeq that can sequence 48 human genomes within 2-3 days. So it's it's the most advanced platform in genomics, and with high fidelity. Besides, we also acquired, because university actually acquired for us also Oxford nanopore technologies. When it's new technology, when you have a pores, biological pore, can sequence your DNA at a long read. So Illumina reads short reads, about 100-150 base pairs, but when Novasec read long reads 2 or 5 kilobase DNA, so it's 5,000 base pairs at or up to 10,000. So because now we can have short and long reads, and then we can easily assemble novel genomes, because this is that's why we're trying now to sequence completely novel, like from marals in east of Kazakhstan, marals and horses of Kazakhstan. And of course there are more projects we have, but I mean but I think we're trying to expand to very interesting areas and of course healthcare is most important. That's why we're sequencing a lot of sequencing genomic projects are focused on healthcare, so working on cardiovascular problems and Alzheimer, and so it means genomics are very support. It's a strong support. We can understand some genomic problems associated with a particular disease, and of course with the Kazakh population. But the most important thing I would like to remember, Iris, so it's because now we are sequence more than 1,000 genomes of Kazakhstani population, mostly Kazakh and other nations in Kazakh. So living Kazakh. So now we are trying to build the Kazakh reference genome. So it means because our genome is definitely distinct, and that's one of the major paper is coming now from National Laboratory of Astana, and of course, as a director, I have to push saying, let's get it to complete this part, because it's very important. Because based on this reference genome, later we can develop personalized medicines. That's why our people, we can actually sequence particular fragments of patients and really compare. Because if you have a reference genome, it will be much better to understand problem associated with this particular patient. So I think this is very critical and important area of research. So, and besides, we also have regenerative medicine, stem cells, microbiomes. It's with bioinformatics, everything is well established. And cancer bio, we have a molecular oncology lab, a cell migration lab. So we have, and it's all Center of Life Sciences. And second center we have is Center of Energy and Advanced Materials. So science. So this center is led by Professor Zhanel Bakenov, and he's also has very strong labs that works on energy, that what's called storage, and then advanced materials, environmental science. So it's I think they're also very strong, and they publish top papers, and I think we are doing good, but I think I still saying we could do better. We could do. I think that's what we're, and of course I think we're getting to the point when we can show and deliver, as I said, top papers for not just in Kazakhstan, but talking very competitive in the world. So marking science, and I think that's, and while we're actually working on this strong, very exciting, strong research programs, we also train a lot of young scientists. So many PhD students, master students, undergraduate students. So and not only at NLA, actually I also have a lab as a biology department. So it means right now we're talking about fusions. That's how we provost other. We always talk how we do all together. I don't think we have any boundaries like NLA or SSH. We just try to build a strong team and deliver science.
Yes. So, so what we've discussed all the time, like in your laboratory, now you are focusing more on healthcare, like solving the healthcare problems related to genome, etc. And you have a lot of kind of departments working, like focusing in a lot of things. And as you're saying, you try to do as much as possible right now and try to give opportunities for youth to like give them more opportunity to try themselves in this new field. Yes.
New field like
No, like in the science field, more like in biology.
no, I mean we're doing high impact research. At the same time, we do a lot of other kind of exciting projects. So, for example, and when people come and they would like to join my lab, I always discuss which area what is interest for you, what is interesting for you, and so. And of course, all my life I study only mammalian cell signaling and cancer bio, like how to drug development, everything. So, and mTOR signaling, of course. But then, and about 3 or 4 years ago, I have a PhD student, actually, Turganbek Khairat, who came and said, I'd like to do, and he's microbiologist. And then I said, dear Turganbek, I have never touched microbes in my life, but then I know you have this background. But let's think about some exciting project together. And what happened is then he was reading a lot of things. We wanted to study how cell growth regulation also takes place in prokaryotes. But later he came up with another project. He said, like, can we study archaea? That archaea, particular that exist in this Kubeituz. Kubeituz is a salt lake that is saturated salt, and there are archaea that live. They, of course, through evolution, adapted to live in this harsh environment. So. We're talking about when we went there 3 times. We had 3 expeditions to Kubeituz. We brought all these samples, and now we established several species, hollow archaea, hollow bacteria that actually grow in these conditions. And we sequence several species of this, and now we don't just don't publish genomes, because we have this ONT technology, Illumina. We have a sequence whole genome of these archaeas, but we realize they are not identical to the because they are subspecies of some other archaea. So, but we want to study how their physiology. So, and based on this, my background, because we study how cells grow, and we know growth is required should depend on nutrients, and now we're trying to understand which nutrients are essential for the survival and growth of this archaea. And now we do a lot of these studies to understand how they what is required, which amino acids or carbohydrates, and so this is another project. I think eventually will lead to very interesting finding, and I think we can later, when we learn the mechanisms how they survive in this and their metabolism pathways, so we can later apply them for develop some new methods in biotechnology, because they adapted to live in this saturated salt. They can grow in foam water salt. Can you imagine if it's almost saturated salt, and then if you put them into physiological normal solutions, they burst within 10 minutes. So it means they actually evolved to live only in this environment. So I think there is in nature there is much more to learn, and that's why my point is we always looking for something interesting and new. And of course, the new members come to the lab. We always discuss and come up with some, I think, exciting projects. So that's one of them. And actually, we have more projects to come. And but of course, at the same time, we would like to complete our high impact projects in drug development, and also we push also basic understanding how ribosomes are built. That's also is continuation of my mTOR signaling pathway study. So technically there is always something exciting you can find, and that's why I love science, because there is always no boundaries. So it's all about your it's kind of commitment, or that's why I always say it's like you have to just love it. If you don't like it, you just don't have to do science. And I always tell to students that science is not for everybody, so you have to really understand if you would like to kind of take this career, you have to just imagine. So you have to only live by science. There is no other way.
Yes, I think that without that inner drive that person must have to do the science, I think you cannot just enforce yourself to do it. Like, I think in that case you'll just go a mess, basically. I think from here we could pass a little bit more about the research related stuff, since we discussed that in NLA and your research about the archaea, etc., and mTOR also. So the signaling pathway, it is much more similar to erasing stuff, actually. I think we could do such analogy. So in a race, when one member like runs a marathon and he passes the baton to another member, and that without this passing, the whole team will fail, basically. And the same could be said about the signaling pathway. So with when a lot of things happen inside the cell, if one thing gets broken or like gets malfunctional, everything falls apart, basically. So could you please describe in a simple way what happens when the signaling pathway does not work properly in our cells?
Your question, actually, it's not as simple as you think. So when you say if you put cell, of course, in a lot of books, explaining cell signals as linear pathway from receptor to goes stepwise, one linear develop. But technically, it's not. It's just explaining how signaling pathway works. But in reality, of course, they intertwine. So, and technically, if you think about it, in the body, of course, all these signaling events happen at the same time, or some of them, so some cells respond to this only signaling. And if actually at the same time, you have to understand that cells, particular mammalian cells, because we went through, it's very complex system. It's if even single cell also have just more limited signaling pathway, but our thing in mammalian system develop into much more diverse and complex system because of different tissues, cell types, and of course. But if you think about, so technically in this system you actually block one signaling pathway, they always go around, find another. So survival, they have actually, as I said, intertwined because deficiency of one particular component might not lead to major problem. But there are crucial points within signals if they're so important. And if you, like, for example, of course, I love mTOR, but if you interfere with mTOR in particular, like, because it has a kinase. So, but we're talking about knockout system right now, when you delete the whole function of this gene, and of course cells will die. So that's why I say eukaryotic cells, there is no life without mTOR, but that's we're talking about central node of signal, because this is critical controller of whole metabolism regulation. And that's why if you delete its function, then cells will not survive. So it means it depends which point of that signaling pathways you actually delete. And at the same time, you have to understand that is because this, every cell has two homologous chromosomes. Even you have a mutation that finds that interfere with the function of this particular gene, there is a second chromosome that you can replace the function and maintain. So technically, its biological system is not, you cannot eliminate just by one hit. So that's what, but if you're lucky, you have to hit something so important. But you imagine there are 20,000 genes, and of course, so far, when we look at, and another thing is, if things happen, if you interfere with essential function, of course, this organism will not develop. That's why if we, when we have these mouse models, we delete essential genes, embryo is not developing. And that's why, I mean, and we cannot even talk about cells. So you delete important factors from a system, there is no organism, there is no life. So the organism cannot develop. Of course, some genes are more essential than others, but we can discuss later. Yeah. So.
Yes. So, as you said, like our organism is not as simple. Like we cannot destroy the whole system by just removing one component.
Yeah.
So, because our like the mammalians, like they are like from the evolution, we are developed to survive, to adapt to changes. So, in this system, I think mTOR protein, which you've been studying like in the USA your whole life, is very important protein that plays important role in proliferation and survival. So can you give like brief explanation of what this protein is and what it controls mostly in our body?
Okay, mTOR is really it's exciting story. So let me tell you how story started. So mTOR didn't come up. So mTOR stands for mechanistics or mammalian target of rapamycin, but usually target of rapamycin. So then, of course, how it started was discovery of compound called rapamycin, but story starts from there was company Wyeth Iris. Now it's called just Wyeth. I think they're in East. It's one of the big pharmaceutical company now. So they realized that because most of the promising drugs are coming from natural compounds, and because these compounds go through evolution, and that's why they are much more stable. They have perfect pharmacokinetics and pharmacodynamics, and usually natural compounds are really become promising drugs. And in the end of 1970s, they started traveling around the world, pick up different soils, and then from these different places, and went back to their labs and the screening was very simple. This piece of soil, and to detect do they have any antibiotic activity or not. So they were particular for eukaryotic system. So they grew yeast Saccharomyces cerevisiae, and if cells did not grow, it means there is something else secreted from the soil that was suppressing growth and of this particular yeast. And then what happened later, they found that soil from Rapa Nui Island, from Caribbean, was really potently suppressing growth of cells. And of course, then they had very strong chemical chemistry department, so they quickly identified this compound that was secreted by some bacteria. It's called Streptomyces hygroscopicus. And this rapamycin was when they quickly resolved the structure, but they knew that now is potent antifungal compound. And when they started pushing it for market, then they realized it's interfering with like dogs have a problem, immune system, T cells drops, and they started saying, oh, maybe it's immunosuppressant drug. And when they start push for immunosuppressants, then they realize it's also suppressing growth of cancer cells. Then they push for as a cancer drug. So, but in reality, it appears to be major regulators of cell growth mechanism. And of course, 1991, then Michael Hall, he's actually very strong geneticist in East, and then he actually quickly did screening, genetic screen, when he actually it was a very strong approach, when effective. So he actually how does screening work? So you take compound and treat all these cells, and then see which cells become resistant to this drug. And then they take these colonies that resistant to the rapamycin, they take the mRNA, make cDNA, and then they express them and to see which cDNA actually responsible for this resistance. And they pull down two different genes. They were very homologous, and they call them target of rapamycin. And it was a Nature paper in 91. And at that time, David Sabatini, as a MD PhD student, and he actually, as a student, imagine as a student, graduate student, he actually he decided to work on rapamycin mammalian system. And then by using pure biochemical approach, because at that we couldn't make the genetic screen in mammalian system. So he could purify this TOR, we call it FRAP at that time. Raft, no, not FRAP, other labs. So because a lot of labs were competing for this discovery, actually, and he actually named it, published Cell paper at that in '94. And that was his major breakthrough, first finding as a student discovering this TOR in mammalian system. So technically, but then later, but we still didn't know how it works. But then we realized that TOR was actually, because of insulin signaling studies, we knew that S6K regulator, phosphorylation, and 4EBP1, and at that time, because TOR was inhibiting protein synthesis, and then they studying these two important regulators for protein synthesis, S6K. It's also called P70 S6 kinase. S6 is a ribosomal protein, but it's a kinase of ribosomal protein S6. So, and then 4EBP1 is also important regulator of translation and protein synthesis, cap-dependent protein synthesis. And then, but still we didn't know the mechanism of work, but we, at that time they were publishing that they probably mTOR directly phosphorylates these two important regulators for protein synthesis. And later move was when David Sabatini received his own lab as a fellow. That's another exciting program at MIT, at Whitehead Institute. When they have very successful young scientists, they actually get a lab immediately after they're receiving PhD. So, and labs, they get some funding to set up their own research program. So, and of course, when he joined Whitehead Institute as a fellow, and he started recruiting scientists, and that's how I got into his lab in '99. So, and of course, our project was to understand how mTOR works, and our task was to purify mTOR interacting protein. That's how you could explain how mTOR works, because most likely mTOR was not functioned by itself. It was a working model. And that's how we were pulling. Our main project was to isolate native mTOR complexes, identify the components. So, no, at that time we thought only one complex, but then we spent all the time. So, and we defined two mTOR complexes, mTOR complex 1, that defined by mTOR interacting with RAPTOR. RAPTOR stands for Regulatory Associated Protein of mTOR. And it's a mTOR, also essential protein. And this mTOR RAPTOR, actually, that's a complex that's nutrient dependent and regulate protein synthesis by directly phosphorylating S6K and 4EBP1. So, and at that time came up with understanding what mTOR is doing. So technically, if we look about, again, complexity, because there was a major jump from prokaryote, archaea, to eukaryotic system, because, and one of the major jump was cell size. If you look about, if you take E. coli cell and average size of mammalian cell, the difference in volume is 1,000 times. 1,000. So it's a major, because even diameter is only 10 times, but if you calculate by volume, it's a major change. And of course, because now, because cell is much bigger, and of course, now it has to monitor nutrients supplies constantly. Because in prokaryotes maybe it's much more simpler, but here it was very critical, because if there are no nutrients, and because we are talking about the rate of metabolism, everything, if there are no nutrients but cells continue to burn its energy and nutrients, then they will die, correct? They'll get into the crisis. So that's why with with this evolutionary jump, with acquiring TOR signaling, because this one cell was pro-eukaryotic cells developed the system, and it appears to be very critical to coordinate life of that cell, because it's based on nutrient availability. And of course, now we know this mTOR is constantly monitoring a level of amino acids available for the cell. And if you look about so for example, we as a humans, when I talk to I say how long you can survive without water, without drinking, and how long you can survive without nutrients, so without food. So, and of course there is a difference, very dramatic. Without water we die very quickly,
Right.
2-3 days.
Yeah, several days.
Because of this any loss of water per cell leading to major stress. But for nutrients, it's different, because as soon cells have sensing that there are no amino acids, level drops, then they switch to frugal economic state, when they burn energy very much slower, and then they actually start turn on autophagy. So that biological role of mTOR, they always coordinate. So it means two mutually exclusive events. So if there are nutrient cells grow, if nutrients drop, then autophagy goes up. But they cannot go at the same time, never, because that's how nature works. So these two mutually exclusive events takes place. And if you think about it, it's now our life is, we have come, most of the I think humans on this planet, they have excessive amount of food. But if you look about evolution, probably sometimes I think tribes of human tribes could be like depleted of nutrients for weeks. I mean, but for 1 week, because they could not catch some the prey and so on. So they could not have access to the food, or there is climate severity and so on. So if you think about this system, of course, just due to of this mTOR signaling, coordination of this rate of metabolism, because of nutrient-dependent metabolism. So I think things where that's why survival, we could survive under extreme life conditions. So it's not only humans; we're talking about all eukaryotic system. I mean, as an example. So, but the only thing I would like to add here is that biological mTOR. But now I think because we have excessive amount of food, and food intake is our problem now. If you look at it, we have hyperactivation of TOR signaling, and that's one of the major problem in the world, with all this diabetes, overweight. It means cardiac problems. And I think it's particularly relevant for Kazakhstan, because I think we have to, as a nation, we have to now, I think, develop some programs like for healthy lifestyle. And healthy lifestyle, what is required is actually proper diet. It means not just you can eat food, whatever, even bishparmak is okay, but you have to eat in the right amount. And of course, exercise, fitness is also critical. So there are two things. And I think if we introduce this right behavior for how food intake and also fitness, I think we can increase just life, prolong our life 20 or 30%, just for basically of this proper food intake and exercise.
Yes, you're right. So when we are going to our relatives, to some kind of events, some family events, we usually bring a lot of food, because it might not be because we want to sometimes provide this food to everyone, but it is also, but I think the main problem is that people think that people might judge them, that they do not put a lot of food. So this might be a kind of consequence of our culture, of our food culture, which you also said about.
No, I mean,
we bring a lot of food, and we eat a lot of food.
I mean, when people, I mean, some occasionally but if you imagine, if you skip lunch, you can have more food, correct? So I'm saying
you're thinking about
it's you have to enjoy. I mean, at the same time, don't have to starve all the time. But the only thing, of course, before at 20, 30 years old, your metabolism is so high. So of course, eating food is not a problem, and even at high amount. So I feel because you are very proactive and your metabolism is high, so you can burn all these calories and you actually move around. You are not life is very active, proactive. But after 40s, I think, at the age of 40, I think metabolism rate drops, and that's why people, start gaining a lot of weight. So it's like you can say so, but some people also, it's not for everyone, but I think I'm talking some people start even gaining weight at much earlier age. So, but it's of course it's important to watch this. Weight is one of those factors that eventually can hit you. Even when you're young, of course, organism very strong. There is everything is can be kind of what's called it can be kind of I mean because resources of organisms, they can actually
Process them.
yeah, they can take it, they can take it, yeah,
Yes.
body can take it, but then of course overweight and so on, but eventually things will go because it cannot tolerate this abuse. Eventually things go down, yeah. So, and of course system is eventually that insulin signaling cannot work very well, and then also blood vessels start build up a lot of so blood vessels get in also in trouble. That's why blood pressure goes up, all these things. So and that's, but if you think about what I'm talking about here is just if proper, you can actually save a lot of even money if you save if you eat properly. I mean, don't to but if you look at our culture, I don't think we go to toy every day. Toys once in a while, and I think we're talking about everyday life. I'm not talking about, of course, if you have go and major occasion toy, it's fine. It's life, correct? You have to get happy. You have to meet your relatives, exciting your friends. I'm not talking about this. I'm talking about everyday life when we also overeat a lot of times. It becomes, I think, my major problem. It becomes habit of eating is because it's like people cannot quit smoking, but if persons start getting used to eating a lot, so it will be habit to continue a lot of eating, and probably that's a problem because it's behavior habit is one of the major things that it's very hard to change. And I don't think it's like culture is. I think culture is great. We meet, and there is another problem. If I think, but if culture is important, if you there was studies that because now major problem in developed countries is Alzheimer, and that's Alzheimer is becomes one of the major problem in developed countries, Japan, US, and so on. And when scientists started to see compare what is they did epidemiological studies, and they came up with very interesting findings. So if you look at US, they have high rate of Alzheimer. But if you look at Mexico, they have low level, much lower. And of course, when they analyze what the data tells them, kind of what is like developing, kind of because they have much less kind of completely different economical state of the country, but they have much less problem with this disease. I mean, they're developing. And when they analyze, and what the answer was, because they have completely different cultural life. So because Mexicans families, they always interact, they go to each other they always have big families that go visit each other, kids come and go. So, and that thing is very critical to maintain your I think brain activity. So because when in elderly people, when there is no social interaction, of course Alzheimer will take over much quicker. Okay, so that's you have to understand that I think social life is very important, and I think what we have in Kazakh culture very rich. I think we have to maintain it. We don't have to sit in our apartments and lock the house and just enjoy, kind of look at the TV. Yeah, so that's I think there is always good thing, and I think human interaction is very important, social life, and that's why we just have to maintain this our society with this culture.
Yes, I think exactly. So even though we're like the same on the inside, on how our bodies operates, etc., the external differences like culture thing also affects us, our health and etc. So even though we're like similar in the inside, our like we have all the same pathway, the same cells and etc. I was curious when you were talking about the mTOR pathway, how it was discovered first, like the effect of rapamycin on, for instance, on cells. At first, you thought that it's immunomodulatory, had immunomodulatory power, but then it was discovered that it also like generally affects the cell growth and the proliferation. So I was curious, how's the understanding of the mTOR pathway difference like now from the past? Do we have like also additional discoveries about the mTOR pathway? What it also regulates besides the cell growth?
No, I mean, growth. That's we can call it mTOR controls anabolic processes. So it's not only, of course, amino acids. There's also building of making fatty acids, cholesterol. They're all connected, and the major discovery was understanding of sensing of amino acids, because at that time when I left David's lab in 2006, of course they continue. I was working on I was talking only complex 1, but then later in 2002 we discovered Raptor, defined functional role, and that was like glue exactly, because that was a gap. So we knew that mTOR works with something, and that phosphorylate S6K4-1 and regulates protein synthesis. And Raptor came exactly to that spot. At that time was critical for us. For the first time we used RNA interference, because it was 2002 techniques to knock down expression of Raptor to show that it has same function as mTOR. But then we found another protein, we call it Richter at the time, rapamycin insensitive companion of mTOR. So this protein appears to be also associated with mTOR, but then Raptor so it's also exclusive factor of binding. So if Raptor mTOR forms complex 1, Richter mTOR form distinct complex called complex 2. But it took for me around 2 years to find its function. Of course, it was function was novel. We knew, because when we discovered it, we knew it doesn't phosphorylate S6K. Didn't do it, and it was when you knock it down, it didn't affect S6K phosphorylation. So it means it was different function. When you knock down Raptor, S6K phosphorylation was going down. So I mean, because it was directly phosphorylating this protein, and it took us a while to understand that it phosphorylates another important kinase, AGC kinase, that controls cell proliferation, and it's deregulated in cancer. So it means it was a very important paper that was published in 2005 when we first discovered Richter, but then we found that it has a major function in phosphorylating AKT, or protein kinase B. This is a very important regulator of cell proliferation that has in growth factor signaling, and it requires 2 phosphorylation events. One was taken by another kinase in growth factor signaling, and second site of regulatory site was phosphorylated by mTOR complex 2. And because everybody were looking for this kinase for many years, for 10 years, but and eventually when we found this, it was a major breakthrough finding, and this paper appeared in 2005 was one of the major that has more than 5,000 citations. I think maybe now it's getting to see. So it's a 2005 paper, but it's I'm saying when you make a major breakthrough finding, of course citation is crazy and also pretty high. So but at the same time you get great satisfaction on that work, and and that's why mTOR now in animal system controls. So let's be complex 1 exists in every eukaryote, from plant, animal cells, everywhere. But complex 2 exists only in animals, from yeast and animal kingdom, not in plants. It was not found. So it means in animal systems, this second complex was evolved only in animals. And this system controls cell proliferation, cell migration, mobility. So it means through phosphorylating different substrates, regulating different important regulators. So And this finding was, that's how we can explain that mTOR is a major regulator of growth of organism, from not only regulating cell size by accumulation of cell mass, but it also controls cell proliferation rate as well through this signal. So that's why mTOR function became much more expanded at that time when I was in David's lab. But then when I left the lab in 2006, I continued working on mTOR complex 2, but David Sabatini defines the mechanism. His lab was still was very productive. I mean, it's so they found that actually sensing mechanism of mTOR depends on lysosomes. Imagine how can you, that lysosomes, it's organelle of degradation, correct? But then now it appears to be during when you activate mTOR after starvation, mTOR complex 1 recruited to the lysosomes by some rep protein, regulatory proteins, that when they recruit it, they actually then they can activate mTOR activity by by directly regulating activity. So technically, this is major breakthrough findings that lysosomes appear to be sensing mechanism of amino acids. So it means, of course, mTOR can degrade some proteins, but at the same time, when amino acids come, they come in and out. So technically, that sensing mechanism now actively studied which amino acids, how they can be work as a sensing mechanism. So I think this is unexpected finding. That's how mTOR is, always brings some exciting new things. At the same time, one thing that we also know pretty well, that mTOR directly regulates rate of building of ribosomes. So whatever we're talking about, it's all happening, translation happens in cytoplasm, but nucleolus, that's where ribosomes getting built, and rate of building of ribosomes determine protein synthesis rate. And of course, when you have to build ribosomes at high rate, and it all happens in nucleolus, in within the nucleus. So, and that organelle is suborganelle is critical, and that's where all this ribosomal DNA that makes ribosomal RNA, they all 200 genes, they all that's where nucleolus formed at that location. And what they do, they transcribe this ribosomal RNA, make ribosomal, and then actually transfer the DNA of ribosomal DNA, and then they make ribosomal RNA that really critical mass for building of ribosomes. Because if you look about ribosomes are ribonucleoprotein, and the ratio of RNA is higher than proteins, although there are about 80 proteins of ribosomal proteins that form a functional ribosome. So that small and large subunit. And but we still do not understand how this mechanism of transcription of ribosomal RNA is regulated. But it's also mTOR dependent. So that's one of the questions we are pushing now in my lab here. We are actually, that's question we actually first characterize pre-ribosomal complexes. It's also a novel finding because pre-ribosomes were never identified, purified, and we developed new methods on, and now we know how to do it, and we are moving now towards understanding how pre-ribosomes get built, and the question is how this ribosome, because there is special ribosomal RNA polymerase 1 that directly controls, that's responsible for expression of ribosomal RNA. So transcribe ribosomal DNA, making ribosomal RNA. So technically, science, I'm saying, never limited. So, and we have this another interesting things, and we know that cancer cells, of course, they overcome, and they actually pushing making much more ribosomes at high rate, and nucleolus. That's why they always have bigger nucleolus, actually, too, because they grow at high rate. So eventually, if we find the particular mechanism, we can actually also block it effectively, and for some cancers, we can actually have a strong impact on growth of cancer cells. But that's, of course, I think one of our priorities understand the mechanism and how they regulate it, what is critical point, and then, of course, we can apply later.
It's actually fascinating that even after so many years after the discovery of mTOR and its pathway, we still need to investigate more on how exactly it operates inside our cells. And several mechanisms are still unknown. As you said, we're still conducting research on them. And as I said, mTOR regulates the protein synthesis inside cells, and proteins are like constitute the vast majority in our body, like I think 60 or 70%. They play a lot of roles in our organisms. And I was just curious, so since mTOR regulates the cell growth, regulates the proteins, could it be somehow implemented to be a target to several diseases, to treat some diseases, like for instance aging or Alzheimer? I know that's broad, sometimes broad, but I think like since mTOR regulates the cell growth, and if we can like stop the cell growth a little bit at least, we can maybe like fight aging or so.
That's exactly. So the point is, I didn't touch this part. We always pushed for accumulated cellular mass, but technically, as we just mentioned, proper diet, actually, of course, now you have proper regulation of mTOR. So hyperactivation leads to obesity, major overweight, and so on. So growth of the organ, because they build a bigger mass, and it is a problem. But of course, now we realize if you inhibit mTOR signaling, but now through, I think diet is the best instead of using rapamycin, but because if you, through proper diet, coordinate your diet, I think we can expand lifetime because we're slowing down aging. So what's happening, our aging clock depends on rate of metabolism. And of course, if your metabolism is at high rate, of course you burn things at metabolism high, so eventually aging also speeding up. And actually, what's happening when overeating leads to higher rate of protein synthesis, and protein synthesis at high rate leads to unfolding protein response, it's stress. And imagine, instead of cell has to facing much more stress under environment that you actually just depends on diet. Of course, too much and particular high caloric diet is also a problem. But here is if proper, of course, you have to eat well, but then if you don't eat also, we cannot just, so there is two extreme things, because some people completely start eating much less, and it's major problem as well. So because food is important, provide energy, building blocks for regeneration. And if some people, because I know there is some people try to go starve for long time. So starvation for, if you don't eat for 1 day, it's perfect, like once per week after 40, 1 day per week, you can just skip breakfast and lunch, it's fine, and you feel much better. And then I think that it's like we're talking about if you skip, just choose 1 day per week, skip this food for, so it means you have you don't eat for whole day from one evening to next evening. So, and that's a really proper way to do it, because when some people start starving for more than a week, 10 days, some 20 days, and eventually it leads to major problem, because when extreme starvation leads to major problem when cells eventually starts eating each other. And when it's happening, some tissues get eaten, degrade much faster than others, and then after long starvation, results into depletion of some tissues. And this is, of course, can lead then a major problem for the organism. So it means everything has to be done in proper balance. And with coordination. But if you read a lot of literature, some doctors even recommend 1 month. They say that, because, of course, for some people it might be okay, but you see, you cannot apply it or recommend it for many people, like for major as a major approach for healthy lifestyle. So I think we have to be careful with this, but I'm talking about 1 day, 2 days, 3 days maximum. But I think it has to be also you have to watch it, and people have to really don't go through the threshold. So but overall, I think if we talk about aging, because I don't think we're ready. Of course, we could take rapamycin, But the problem, rapamycin is very perfect target. It's not directly, it's allosteric inhibitor of TOR, mTOR complex 1. So, because complex 2 cannot be targeted by this drug. But here, inhibition of mTOR actually leads, because as I mentioned at the beginning, it leads to suppression of immune system. T cells become much less, small number, because it blocks proliferation T cells. And eventually, of course, immune system weakens. And when now there are several publications took place in Nature, and so they say that mouse can survive longer when they are treated by rapamycin after because they usually live 2 years in labs, but when they starting after 18 months, they treat mouse rapamycin or 1 year, and they can live longer. But because now we have to consider this, mouse they live in a clean environment,
yes.
and of course they slow down the metabolism through rapamycin specifically, and because they're not facing our pathogens, and that's why they can live longer. But I don't think we can apply this system using rapamycin to prolong life for humans. The only thing, the best, I think, common sense. That's I always say. Common sense is with the knowledge we have. I think proper balancing of mTOR and autophagy. So I think skipping meals once in a while, even for whole day, is okay, but don't go over than 1 week or so. This is really will be bad. And another thing, of course, fitness, because at the same time you have to maintain your muscle through, because when people are not going to any physical work, then their muscles get degraded as well. So and I think losing the muscle mass is also a problem. It leads to eventually a problem in your life. So, and of course, and be happy as well. Those important. So you have to be open. I mean, psychologically also, it's like find some happiness in your life, to do something exciting, and that I think will be also critical.
And I think on this positive note, we can go to the conclusion section of our podcast. So, Balkiya, do you have anything to say?
Yes,
I think I'll add up later.
So, as a second-year biology student, I may not understand some of the details of the discussion, but sure, I understand most of the part, like how the mechanism of cell signaling works, also how the drug works, like the mechanism of the combination of vitamin C and arsenic, like Mishiak, as you said, and it's really interesting that how those ideas came up together and gave this product as a result. So you said that you also met a lot of people along your journey, and it was really kind of productive talk. And we learned also more about the National Laboratory of Astana, in which you are a director, and about what kind of work is done in this institution. So I think it will be very useful also for our listeners, maybe who, I think there are a lot of students, people who will be really interested in joining your laboratory and your research group. And we are really thankful that you joined us today and shared a lot of insights with us.
Thank you very much. Yeah, I mean, young scientists, always students and researchers are always welcome. And I think I'll also look forward. And I think right now we're in a critical stage, and science can have a high impact, but you have to do it right. So we sometimes have to take our pace and then do it correct. We cannot rush our clinical trials. So, and just a couple of words of clinical trials. So first dosage has to be right, defined maximum tolerable dosage. And second thing, because cancers are highly diverse, and we know the KRAS mutant, but even KRAS mutations can be different, and And there are a lot of other type of cancers that could be also they also highly addictive to glucose. So eventually, through clinical trial phase 1 and phase 2, we have to define this type of cancer, but this is can be defined through genomic profiling, because through genomic profile we can see exactly all the mutations, oncogenic factors, mutation of tumor suppressor, and how mutation oncogenes. So define oncogenes, and then we can say exactly what type of cancer will be sensitive and not sensitive to this drug treatment. So it means it's work in progress. And I hope I have had very great conversation, and I'll be always happy to provide more details. And Kob Rahmet.
Yes. So thank you very much for joining us today. And we hope that our viewers, our listeners have found this episode engaging and that with new insights on the cancer, on the chemical pathways, signaling pathway in our body. So please don't forget to subscribe so you don't miss our upcoming episodes. And see you later.
Goodbye.
Bye. Kob Rahmet. Thank you.